Okay. Okay. So hello everyone. My name is Alitzel López. I'm a first-year PhD student at the University of Sherbrooke, and today I'll be talking about colorful orthology clusters. So I'm sure you're intrigued by the word colorful. So mostly because I am Mexican, I used to see things in color. Well, that's one reason, but the main reason comes from homology. So homology is what we use to study evolution, and we use this through species trees and gene trees, which are just hypotheses of the evolutions of the species or of the genes. So in gene trees, we can find two interesting evolutionary events, which are speciations and duplications. And these events actually yield many relationships between the genes that which are involved in a study. When two genes have a last common ancestor, that is an speciation event, we call them orthologs. And when they have a last common ancestor, which is a duplication event, we call them paralogs. Orthologs are particularly interesting because they can help us reconstruct species and gene trees, and they also help us predict gene functionality. They are also like the protagonist of the ortholog conjecture, which states that orthologs share function while paralogs diverge. So throughout the literature, we are going to find two different methods for orthology inference. First of all, we have the phylogeny-based methods, which explicitly reconstruct these two trees, so species trees and gene trees, and try to overlap them and explain the differences between them through speciation and duplication events. And on the other hand, we have similarity-based methods, also called graph-based methods, which will be the main focus of my talk. So in similarity-based methods, what we do is that we make pairwise comparisons through the assumption that sequence similarity is a measure of evolutionary closeness. So you run this to, for example, a BLAST, an all versus all genome. So we have, for example, the pig genome and the rooster genome, and we compare each of their genes. And whenever we have a reciprocal hit, we say that they're most similar. So with these pairwise comparisons, we build something called a similarity graph, where this is a mathematical structure in which the colors represent the species and the vertices, they represent the genes to which they belong. And the edges represent these evolutionary relationships. So when we finish building the similarity graph, the next step is to make a cluster to find the clusters of orthologs, which will be the main topic of my talk. So most of orthology inference tools actually differ from this clustering step. So for example, OrthoMCL uses Markov clustering, which simulates random walks through the graph, while the OMA database, they use what is called hierarchical orthologs. So they study clusters within a specific given taxonomic range. And protein ortho, which is the fastest of these methods, uses something called spectral clustering, which focuses on the algebraic connectivity of the graph, and it uses the adjacency matrix of the given similarity graph. So although these methods are faster than phylogeny-based methods, they have many important drawbacks, and they struggle whenever this scenario happens. So whenever we have an speciation, which is followed by a duplication event, this creates imparalogs. So whenever we have candidate clusters that look like the ones on my left, these candidate clusters actually either they could contain many missing edges, and most importantly, they also contain some paralog genes. So the methods that I mentioned before, they do not actually consider the precedence of each of the genes. That is, they do not take the colors into consideration. So whenever I say colorful, I want to refer that each vertex has a different color. So in this work, what we do is that we study these colorful clusters. So clusters where each vertex has a different color. And we combined our method of inferring colorful clusters with another method that helps us infer the intercluster orthologs. So the advantages of this is that we get clusters that have purely orthologs, and this helps us better reconstruct the evolutionary histories. They also help us to distinguish orthologs that have diverged following a duplication event. So the main motivation of this work is that when we have orthology inference studies, usually the number of species that we consider is small, and also the number of potential orthologs that we consider for each species is small. So we try to devise an analytical way of combining these two constraints into a parameter, and through the help of a mathematical structure that we call the best neighbor graphs, we study the problem of clustering by bounding the number of colors and neighbors of each color that each gene can have. We also obtain some hardness results, a fixed parameter tractable algorithm, and we illustrate the usefulness of our method by doing experiments on simulated data. So here is our mathematical structure. So the best neighbor graph is a color graph. It's a properly colored graph. This means that no two endpoints have the same color, and where each vertex has at most d neighbors of the same color. So with this structure, we formulate a computational problem as a colorful clustering problem. That is, if I give you a KD best neighbor graph and a cost function, that is, this cost function represents the number of edges that I have to add or delete, so editing operations that I have to do to this graph, so that we get a partition into colorful subsets, that is where each subset has exactly one vertex of each color. That minimizes the cost function. So what can we use as a cost function? So in computer science, there is the classical cluster editing cost, which is here. So suppose that we have a candidate cluster like this in gray, and to calculate the cluster editing cost, classically, we will just count the number of edges that we will have to delete, for example, this one, and the number of edges that we have to add to make this a complete, to make this a click. So where every component is a neighbor with the other ones. Every node is a neighbor, has all of its neighbors, everything is connected. So we take the classical cluster editing cost and we make a colorful remake. This is, we want to have a cluster editing cost that also considers the colors. So for example, we see that the previous cost function actually over-penalizes some edges. In here, we want to penalize only the colors that we are not considering into the cluster. For example, we see that the colors in the candidate cluster are these ones, and the only color that we will be missing, so the color that is not present in the cluster, will be the purple one. So we will be over-penalized. If we take the classical cluster editing cost, we will be over-penalizing this edge, which is already represented here. So this node already has a potential ortholog of the species, so we don't want to penalize this edge. But in this case, for example, the purple edge is missing, so we want to penalize this color that is missing from the cluster. This idea is represented in the first term of our editing cost, and in the second term, we just penalize the number of edges that we should add for it to be a complete, for it to be a click. So with this, we present some theoretical results. We ask some questions, and we want to know whether or not these questions are difficult or not. So to which graph can we apply K colors to get a K1 best neighbor graph? This question is easy whenever we have less than 3 colors, and it becomes hard whenever we have more than 4 colors. And also we ask ourselves, is colorful clustering K1 best neighbor graphs easy? We see that this is an NP-hard question whenever we have 37 or more colors. And we provide an algorithm for finding one most modular cluster in FPT time. So FPT time, for practical purposes, means that it's an algorithm that you cannot run when the deadline for the conference is due tomorrow. So because it has an exponential time component, so this exponential time component will remain exponential, but is bounded by the number of neighbors and colors that we studied as parameters. So to illustrate our method, the usefulness of our method, we made some simulations using a tool called Asymmetry, where we simulate also a noisy distance factor. So we have some noise factor, which will represent the experimental noise that we can have in the measurements. We build the similarity and the best neighbor graphs from the simulations. And we compare our method with spectral clustering, which is the clustering method used by protein ortho. So how do we, well, our experimental setting is like this. We have our ground truth, which will be the true gene trees generated by asymmetry. And from these true gene trees, we have a true orthology graph. So a graph that represents all the correct orthology relations. So we will say that an edge is a true positive whenever we infer the correct orthology relations. We will have a false positive whenever we have a paralogy that is interpreted as an orthology, and we will have false negatives whenever we have orthology relations that are left undetermined or that are predicted as paralogy. So we see in the experimental results that our method remains competitive with respect to spectral clustering. So spectral clustering is SPC, and our method is MMC. So we see that precision is greater than recall in most of our experiments. So this means that the found orthologs are correct, but we have many orthologs that are missed. We also combine our method with another, as I said, with another method that helps us recover the intercluster orthologs, which is called HIPPO, so hybrid prediction of orthologs. And we see that in the presence of moderate noise, our method remains competitive. And also when we increase the noise to, we have a noise factor of 2, we see that our method still remains competitive. So unfortunately, one of the limitations of our model is the exponential complexity. So the number of species that we can handle now, it's limited. However, we have new ideas for future work. That is, for example, testing our method against other orthology tools, not only protein ortho, but also we have the idea of adapting fast algorithms such as spectral clustering to colorful versions. So to something that actually considers the precedence of each gene. And without further ado, I thank you for your attention.
We have time for one quick question.
Yes.
So the exponential time of the order, what is the factor that's exponential? Is it the number of species?
No, it's the number of. So since we have all the, we look for all the possible subsets of. Well, there is one constant which I don't remember, but the exponential part is it's a sum between the number of potential neighbors and the number of potential orthologs. Yes, we have a constant, and it's to the power of the number of potential orthologs and number of neighbors. Yes, colors and potential neighbors. Yes.
So you talked about the FPT, but my question about that is it possible to consider small instances of problems, solve the problem, use algorithms to solve the problem on load, and then merge the solution together?
I think it would be possible. I think I will need more time to answer that question, but I think it can be possible. Yes. Yes, of course. Yeah. Thank you.